and welcome to our Ocean Optimism Speaker Series. My name is Emily Cook. I'm the Manager of Programs here at the Nicholas Sonntag Marine Education Center, and I'll be your host tonight. We're so excited to have Sheila Byers joining us to talk about glass sponge reefs and bottom contact fishing in the Howe Sound. Before I bring Sheila on, I'm just going to go over a few things. So I wanted to start off with a land acknowledgement. So the Nicholas Sonntag Marine Education Center acknowledges with respect that our work takes place on the unceded territories of the Squamish Nation. And as this is a virtual event, we implore all of you as participants to do some research into the Indigenous territories that you are currently living on and occupying. And if you feel like putting that into the chat so that we can get a sense of where you're joining us from, that's always appreciated. And then as we're on Zoom tonight, a few etiquettes to go over. Just make sure that you keep yourself muted through the speaker through the event so that people can hear the speaker clearly. If you also don't mind turning off your camera so that everyone can kind of focus on the speaker themselves. We'll have a question period at the end of the talk, so you can type your questions into the chat, and we'll have some time to read those out to Sheila. And then just to note that this talk is being recorded tonight, and we'll be making it available on our YouTube channel and website coming up in the next week or so. I'd really like to thank our presenting sponsor, the Sunshine Coast Foundation, that has made this series possible. This is our fourth and final event this winter, and they have supported us in making sure that we've been able to support each speaker financially to present, and just make sure that our staff hours and all of that is covered. So we're really appreciative to the Sunshine Coast Foundation for that. I also want to thank our board of directors and our technical advisory committee who worked with our staff to kind of work on the theme of the event and to find our speakers and bring them together to make this event possible. So without further ado, I would love to introduce you to Sheila Byers. Sheila is the Vice President of the Marine Life Sanctuaries Society. She's also the Curatorial Assistant of the Marine Invertebrate Collection at the Beaty Biodiversity Museum. And Sheila's worked very closely with glass sponge reefs and done research and has so much knowledge that I'm very excited to learn from her tonight. So without further ado, Sheila Byers. And Sheila, I'm just going to stop sharing my screen and then you can start sharing yours.
Okay, thank you.
Okay, so you should be good to go now.
And there we are, and there we are. Okay, hopefully everyone can see that. I'm just going to pull up the laser pointer here. So thank you very much, Emily. That was very nice to be here. Thank you so much for the invitation. And I think I owe some of this to Colin Levings, who's online here as well. Colin, I've known for a while. But I would like to, I appreciate very much having the opportunity here, being as Gibson's is so much a part of Howe Sound. I would like to acknowledge that I am living on the ancestral, traditional, and unceded territories of the Tsleil-Waututh, the Squamish, and the Musqueam peoples. And I have to say that I wake up every day loving nature around me, the beauty of it, and how empowering it is with its resiliency. And I would like to thank the coastal nations for the opportunity to share this land with them. Okay, so we are going to look at glass sponge reefs, primarily in Howe Sound, but I'll do a little bit more of a broader view initially. I always like to have this slide in by Bettina Matsgoon, who did a beautiful, beautiful and quite a large embroidery piece on the what she called the City of Glass. I worked with her through another society that I belong to called Nature Vancouver. When it was first shown, it was gone that same evening. It just gives an idea of how intricate her work is, but more to the point, it's lovely to bring art and science together. So I'm going to try and do this in a nutshell, although it looks like I'm covering a lot. I am. But we're going to have a look at what sponges are, let alone glass sponges, what glass sponge reefs are all about and how they work, why they are unique and special from what we would call an ecological perspective. I'll talk a little bit about the citizen science work that has been done by Marine Life Sanctuaries over the past number of decades, why we consider these sensitive benthic habitats, and then talk a bit more about marine refuges and bottom contact fishing. So hold on, here we go. First of all, sponges. You may know sponges by your bath sponge, although they're probably not real anymore, more synthetic, but they do demonstrate one of the 4 major groups of sponges. Demospongia, I don't expect you to remember any of this, but it's just something that you probably recognize. They have lots of holes, as you can see here. So that's what porifera means, pore bearers. And their structure, what there is of it, although many of you who have had a bath sponge know that it's quite flexible, it's made of a type of protein. So 90% of all of the sponges in the world are this type of sponge. But I really want to focus in on the hexactinella, or the true glass sponges. They literally are made of silicon dioxide, or what I'm going to refer to primarily as silica. Now, there are 2 other groups that have a funny combination of the protein spongin and the glass, and also calcium, if you think of shells. So in combination with those other 2 groups, the glass sponges are comprising less than 10% of all the sponges in the world. So right off the bat, we get to know that glass sponges are rare. And in fact, at this point in time, the west coast of North America, primarily BC, is the only place that these reefs are found. But before I get into too much more about reefs, here are 2 glass sponges. Okay, they are different species. One's called the boot sponge, one's called the fingered goblet. You can see some fingers coming down here, but they are not both of a type that builds reefs. This one builds reefs, this one does not. So what's the difference? Well, they're made up of, or their skeleton is made up of silica, and they are 6-pointed like this. But you can look at this graph or image here and notice the many varieties of different types of spicules. So those will vary by species. But more importantly, that boot sponge is going to have these spicules arranged in a bit of a disorderly manner, something like that, or maybe like this. Whereas that fingered goblet sponge, the one that does contribute to building reefs, looks more like this. So you could almost think of scaffolding that's put up around a new building. So the tips of these 6-pointed spicules are fused, giving it a much more structured formation, the skeleton. Okay, so, and you should also think that these are actually coming from igneous rock, or igneous rock that's being broken down into granite, quartz, sand, over a long period of time of freezing and thawing of, guess what, our coastal mountains, which go all the way up to, and I'm not sure if they go into Alaska or not, but they certainly travel from here all the way up the coast. And that's where a lot of that silicon dioxide, the dissolved form of silica, is originating. Okay, now just to try and give you an idea of how these sponges work. So we're going to start with a bath sponge. This is a very simple drawing, but take note of the wall of the sponge here. And if you look closely, you can see these tiny little spicules. And that skeleton or scaffolding is completely surrounded and infused with tissue. Okay, so the living part of the sponge is being held up by that rigid skeleton. You can see holes here. Remember, porifera is pore bearing. So all of these holes, like here and here and here, water is being pulled from the outside in through that wall through a very direct channel into this inner, generally wider opening called an osculum. Now, you'll notice these funny little cells on the inside wall here that are enlarged to look at this. I'm going to call them a collar cell because they have a collar like this that's full of mucus, but they also have this flagellum or whip-like structure that is pumping and working hard to pull the water from the outside of the sponge in through these pores and out through that main osculum. So that's really important in creating that movement of water coming in, because that's how the sponge is getting its oxygen and food. And then on the way out, it's dumping off its carbon dioxide and other waste materials. So that's kind of a basic structure of the bath sponge and how it functions. So if we go to a glass sponge, there's another generalized but a little bit more complicated image to show something like the fingered goblet that we were seeing. But you'll notice here that the pores do not, so this is the wall again, but the pores do not go straight through like they did in those bath sponges. Instead, they go through smaller and smaller and smaller channels before coming out through another exit into that osculum, or central opening. So right off the bat, again, not only are they rare, but they have a very complex channeling system that gets complicated when we start talking about food and movement of water through the wall of these glass sponges. What do they like and extract mostly out of the water is its bacteria or tiny little types of plankton. Hopefully you can see from this image that because of this complicated channeling system, there is potential for material, organic material that's coming in through the walls, the pore in the wall, that they could clog these small channels if they were too big. If the particles of that organic matter or sediments was too big, it could easily clog them, and meaning that they have to stop so that they can try and get rid of the clogging, or potentially it could lead to death. I want to take a quick note here of Dr. Sally Lees, who has led an awful lot of the research on sponges on the coast. She did her PhD at the University of Victoria and has been working at the University of Alberta actually for a number of decades, I believe. And many of her students are well known for their work on coastal sponges. And Dr. Bill Austin is another person. Unfortunately, he did pass away a few number of years ago, but had a lot of important contributions to make to the sponges that were being newly found on the West Coast. Okay, so I noted that the fingered goblet sponge was one of the types, one of the species of sponges that actually forms reefs. So it has a very structured internal skeleton of fused spicules. Another one is what we call, and sorry, you can see the fingers coming down here. Sometimes they're vase shaped, but definitely lots of fingers here and tubes. There's your osculum here, whoops. And then this more yellow type of species, a cloud sponge. And you can imagine large fluffy, what is it, columnus clouds? Oh, forget that. I like to call them the mitten sponge because it kind of reminds me with big mittens here. There is a third species. This one is a lace. That's a very tentative common name, but a lace sponge, but you can see it is much more delicate looking. All three of these have been found to build the reefs that have been discovered in British Columbia. I'm going to note, however, that these two species, the cloud sponge and the fingered goblet, are only found in the Salish Sea, Strait of Georgia and Howe Sound. This one does not occur in the more southern area. I want you to pay particular consideration to the fingers and the mittens in the sense that when water is moving over these reefs, and mind you, you're only seeing a very small section of a reef here, that water bringing organic material, food, sediments, gets baffled by these structures such that the organic material and sediments will fall to the bottom to form this very silty clay-ish substrate. And why that is important is that, whoops, I'll get to it in the next slide. Where were they first found? The reefs in the Hecate Strait and Queen Charlotte Sound area. So you can see three very large reefs here. This was in about 1986-87, primarily with Dr. Kim Conway as the with the Geological Society of Canada. He was mapping all the way up and down the coast and came across weird structures that he was not able to put his finger on. And so they had to get a submersible out and have a look. Note that when they were founded, or sorry, when they were discovered and then became part of an Oceans Act, sorry, I have to move this away, was not until 2017. That's a really long time, but do know that not a lot of information was known about these in this early decades, even into the 90s. So what did they find? What was this weird structure that they were finding? It looked something like this. So you've got a sponge reef foundation or structure with live sponges growing on the top. So that means that when one generation of sponges dies naturally or otherwise, that firm skeletal structure collapses. So that means that all of that tissue is going to decay. But don't forget the baffling that the cloud sponge and the fingered goblet sponge were doing with the sediments. That has already fallen to the seafloor. They sort of combine over time to form a firm structure or basis on which the next generation of sponges will settle. So time after time, as the older sponges die and collapse with the sediment, that reef gets higher. In fact, it can get as high as 25 meters and kilometers in area, as you can see by the area encompassed in this particular image. Notice that the new sponge are on top of that those previous generations, the substrate. They can grow as high as 1 to 2 meters. So we, and generally speaking, new larval sponges settle on the dead previous generations that we'll be showing through, depending on the concentration of the live sponges. So we're talking really deep, really dark, really cold, so less than 12 degrees centigrade. And sponge reefs so far are anywhere from about 40 meters to 240 meters. So definitely the ones in Hecate Strait, Queen Charlotte Sound were really deep and on the continental shelf. Okay. One of the things that we think is a reason why they're found, that their reefs are primarily found on ridges or pinnacles or seamounts, is that they need really high tidal currents or movement of water that's bringing them the organic matter, which will be containing their food, that is bacteria, but also the sediments to help build the structure. So we think that that's maybe one of the reasons why they're found elevated as opposed to on the flat seafloor bottom, or the seabed, I should say. Hecate Strait, Queen Charlotte Sound was a little bit different because it was wide open to the Pacific Ocean coming in through there with lots of well oxygenated and carrying lots of food in those currents. Okay, so yes, they're needing to baffle the sediments to extract the organic sediments, that's going to hold their bacteria, to get the silica, as in dissolved silicon dioxide, out so that they can actually build those spicules to build their skeletons. And bacteria, I said that twice. Okay, hopefully that kind of gives you an idea from a cross view of what these reefs are looking like. Okay, so we can talk about ecological values. So how are the sponges and the sponge reefs interacting with their environment? We can call them values, we can call them benefits or services. And I will say that these words are meaning, how do they benefit us? So we can look at benthic pelagic coupling, which means connecting with the plankton, and looking at the reefs as foundation ecosystems, because in these deep sea environments, cold, dark, there's often not a lot of food for other types of marine life to feed on, but more importantly, structures that will actually allow some of these marine, different types of marine organisms to find hiding areas or to get more food. So let's look at both of those. Okay, back to our two species of sponge, the cloud sponge and figured goblet sponge. You saw those little whips of the collar cells and how they're going to be beating away, pulling the water from the outside in through the walls and the skeleton of the sponges out through the central pores. That is huge, not only hugely important, but immensely incredible for what we often consider very primitive marine animals. In fact, of all of the, I'll say, ecosystems or communities that are known from the ocean, these sponges and the sponge reefs have the highest known transfer of water from the bottom of the ocean. That's why they're called benthic, they live on the bottom, to the pelagic area where plankton live. Okay, so that's a huge transfer of water, pumping all those little collar cells, pumping away, pulling in water that is actually coming from the pelagic upper reaches of the ocean through their skeleton, out and back up to the surface waters again. So what that means is that the sponges indirectly are supporting the pelagic planktonic community, because all of that waste material that they've disposed of coming out through the osculum is now being transported up to the surface waters, which will actually help to provide the necessary nutrients for plankton to bloom. So that's a really big deal. So big that that function of the collar cells and their little whips and their busy pumping activity actually amounts to about 6,500 Olympic-sized pools daily in Howe Sound. That's 17 billion liters per day. Or we can look at it a little bit differently, because there are reefs in the Strait of Georgia. I'm going to show you those shortly, as well as in Howe Sound. And if we put all of that pumping together, they require, or in 82 days, they can filter all of the water in this area, which means that 4 times a year, water is being exchanged from bottom to top by these glass sponge reefs. That's huge, huge. In terms of the structure, all of that organic material from the tissue of the sponges, not to mention all the organic material that may have fallen out of the water as it's the tidal current, as it's being baffled by the sponges, falls to the bottom and becomes part of the structure. Don't worry so much about this, but it is good to know that the amount of carbon that these reefs capture and sequester, as in like a sink, is very close, if not equivalent, to the terrestrial old growth forests and giant kelp forests. So that also is another amazing factor or feature of these sponges. Whoops, and I missed silica. So that's not too hard to visualize. When the skeletons, when the sponges die and collapse of one generation, all of that silica and the tissue is going to decompose, and so you're going to get a sequestration of silica too. This becomes particularly important when we start thinking about climate change. Okay, so we see here in about 2002, 2010, again, Kim Conway and his crew were mapping this area and found these sponge reefs. Sorry, I should start down there, up here, here, and into the sea shelf area. They also found a reef here, as well as at the upper end of Howe Sound, more towards Squamish. So they received fisheries closures in 2015, 2016. So again, that's a much better time frame than we saw for the Hecate Strait reefs. But of course, they were the first ones to be found. There was lots to learn by even this date. I'll introduce you to Marine Life Sanctuaries Society, because this is the society that I'm a part of, and that our focus is on Howe Sound, at least for the time being. But it was first started by co-founders, Andy Lamb and Bernie Hanby, which some of you may know from the Marine Life of the Pacific Northwest, that beautiful big green book of photographs. They and a number of other people who founded the society were focusing on rockfish conservations. They were all divers, and they could tell over their decades of diving, even back in 1990, that rockfish were declining. So that was the initial focus of the society. But when DFO, Fisheries and Oceans Canada, start to be concerned about protecting not just individual species, but recognizing the importance of reefs, of ecosystems like the glass sponge reef, then our focus became more directed to the actual overall reef ecosystem. So we do a lot of citizen science work. We're all volunteers, and things like mapping sponge reefs. We do top-down, or that often involves, because this is done by boat, top-down drop camera. So a camera gear is being lowered over the side of the vessel, and videos taken from above the reefs and the sponges. But we also have diver perspectives in photography and videography, as well as temperature logging and some other field experiments. I'm not going to go into too much detail on that, but the reason that we are able getting ahead of myself, these are in red, the 12 locations that Glenn in particular, here's Glenn here, and this is his Skyliner vessel that we have used to do a lot of this diving. He knew, or he recognized glass sponges from early dives as far back as 1984, but he did not really understand the significance or the fact that they formed reefs, probably until into the 90s. And then really, when he realized that and knew that there were many more reefs in Howe Sound than had previously been discovered by Kim Conway and his group, he started to do the mapping and drop-down camera work himself out of his vessel. And one of the very unique things about the glass sponge reefs in Howe Sound is that 5 of them are actually air diveable. This is unique in the world. So as shallow as 40 meters, we're still getting down, depending on the tidal cycle, to 75 over 100 feet. I'm going to try and stay with meters, but nonetheless, it is diveable, air diveable. It's still tricky, and you don't get a lot of bottom time, and you need to be an experienced diver, but nonetheless, it makes these reefs incredibly valuable to conduct research, and just for people to be recreational divers to be awed and amazed by what they're seeing. So this is one of the maps that Glenn would have created. Here is the southeast corner of Gambier Island. Here is where the Halkett Reef is. So red is actually shallower. And this is the sort of information that he came up and was able to have written up in Canadian Field Naturalist Journal. That was in 2017, and also another report that Laura Trione of the Society had contributed to DFO. So in other words, all of this information has gone to DFO and helped them in their effort to provide evidence of the value and importance of these reefs. So ground truthing is required by DFO on any of this sort of information, and they would use a ROV, an ROV, remotely operated vehicle. And this is the sort of view that they would get. This is not unsimilar to what the drop camera used by Glenn would see. You're looking down on top of these sponges, keeping in mind that they could be as high as 2 meters. And you can see the dead generation of sponges behind or below, sorry. DFO, when they used the remotely operated vehicles, were able to identify about 84 taxa, or groups of different organisms, including fish and invertebrates, that sort of thing. Even looking down from the top, they were able to gather that much information. But MLSS also had that advantage of divers who are getting a much closer, what I like to refer to as a fisheye view, face to face, right down on the bottom, because as you can see, there's lots of little holes and crevices that there may be lots of things hiding. You're not going to see it with a top-down image. You need to be right on the ground, so to speak, to look directly diver view. So, oh, did I skip something? No, okay, sorry. Okay, so that's what you would see in the sense of a bigger picture of these reefs. We need to look a little bit closer at the values that a smaller picture or close-up view would give us. What are the benefits of glass sponge reefs to other marine life? So I'm going to go through this a little bit faster. These are the sorts of views that divers get and what they see. You need to know that rockfish are one of the dominant species that are associated with glass sponge reefs. This is important because these were the fish that Andy and Bernie were concerned about because their numbers and their populations were declining. Rockfish are long-living, very long-living, and they, unlike many other species of fish, give live birth. So finding out what was happening within the reef, what became a really important and informative aspect of diving. You can see from this quillback that she is pregnant. Okay, so that means, are they using it as a nursery? Well, we're going to find out. This is a yelloweye rockfish, also found amongst these sponges and reefs. This is a subadult yelloweye here, also a very long-lived rockfish. When I say long-lived, I need you to think about, let's use 100 years as an average lifespan. That's not uncommon. They mature, say, 15 to 20 years old. So if these fish are caught, that means that there's at least 80 to 85 years where you have lost the next generations of rockfish. And that becomes more important when you start to think about, unlike humans, where females, our eggs get not so great the older we get. For the female rockfish, the older they are, the more viable those eggs are and the better chance they have of surviving. So you can see why Andy and Bernie were so concerned. If you're taking out that many generations, the populations are going to really decline rapidly. Okay, now this was a very exciting moment for Adam Taylor, who is one of our photographers and one of our board members actually, to photograph a young of the year yelloweye rockfish. This is very tiny. This is showing that, yes, indeed, rockfish are using these sponges as nursery grounds. And you can imagine there's lots of cubby holes for small juvenile fish to hide and be protected. Remember, these are islands in the middle of this vast ocean seabed with little place to hide. And then you've got these incredible reefs that are providing hiding places, nursery grounds. Okay, this was another really important moment. Diane Reed photographed a pregnant Puget Sound rockfish just about just about to give birth. And in fact, she caught the flap open so that the live baby rockfish are about to drop out, sorry. So again, you can see the relevance of all of these areas for hiding these young larval rockfish in particular, where they can grow up, find food, hopefully, and become adults. Okay, so I should say that rockfish are an important recreational fish, and lingcod, that's both recreational and commercial fishery, not in Howe Sound. This is an eggs case. No, it's not a case. Egg. Anyway, eggs. There's another term that I'm forgetting. Laid by the lingcod, and this is the male looking after them. Very protective and aggressive in that moment. There are things like female greenlings, kelp greenlings here. There's Pacific octopus, again, both recreational type of fishes. Tanner crabs. Here we have a larger male with holding on to the female where they will mate. So obviously also making use of this protective area for mating. Things like longhorn decorated crabs find refuge in these tiny little, or in these openings, the osculum of the sponges. And if we keep going down, we will get to things like squat lobsters. They're quite funny little, not lobsters really, they're more closely related to crabs, but nonetheless, many, many, many of them. Notice this little new sponge that the larvae would have landed on the dead sponge because it's brown. There's no white tissue there like there is here of the live sponge. And another very young new glass sponge that is settled here. Things like shrimp are also found in the reefs. This one still remains unidentified, I believe. Even smaller, this isopod. So if anybody has seen the little pill bugs in your backyard, roly-polies, this is a saltwater relative. So they're all around these sponges. This one's going to be a challenge. We have squat lobsters, but there's something else in blue here that I'm hoping you can see and that I can find right here. A copepod. There's another one there. I know there's a few more around, but you can see that they're smaller. So the idea here is that we've gone basically from large to small, just as we might think of a food web with a lot of small organisms like the copepods that things like shrimp and squat lobsters would survive on. And then the squat lobsters are probably getting eaten by fish or maybe even crabs. So in other words, the reef is providing a food web for these organisms, and that's what is making that ecosystem, that entire reef, so important to the marine life in the ocean. Okay, so from that marine science, citizen science perspective, 5 air diveable reefs allowed us to find marine life supported by these, what I call islands. We found evidence of reefs as a foundation species, providing important habitat for nurseries, foraging grounds, and refuge for a lot of other marine life. And certainly the fishes and the crabs, evidence of commercial, recreational, and cultural species are thriving in these ecosystems. But there was a missing link. That's the larval form of the glass sponge. Where is it? Where is it coming from? Is it landing on these reefs in Howe Sound? We knew that the evidence of larvae had been found in the Strait of Georgia, but so far not in Howe Sound. So we set about doing that with what we call the Larval Settlement Plate Project. As with the drop camera and most of the gear that Glenn uses, this was designed and built by him with contributions from MLSS, other MLSS members, and a DFO. It's a tall tree with plates, and that was installed in March of 2017. You can see the divers here and the tree having been installed. I'm not going to go into any detail there, but there are 2 plates there, 2 plates there, and 4 on the lower branch. And that's, again, the importance of having that diver ability to go down and photograph these approximately every 3 months, the plates. What did we find? Well, here's an example of one of the ceramic plates. We used 2 types, ceramic and glass. After 3 months, they kind of looked like this. Notice colonies here. After 13 months, we were seeing more adult-like invertebrates, of which these all are. Here's some tube worms, and here's something called a tunicate, or you may be familiar with sea peaches. They are similar, belong to the similar group. And we got 70, or we discerned 70 different taxa or groups of marine invertebrates in particular on these plates. So don't worry about the complexity of this, just notice red, majority of taxa that were found, 9 to 29, higher, such that as we move down the page, we are coming to fewer taxa, but nonetheless, worms, moss animals, hydroids, which are relatives of sea jellies, and bivalves, gastropods, so shells, basically. Okay, so in other words, 4 dominant groups were found, things like worms, found and that we could identify either as still very young juveniles or adults, but if they were there long enough to grow into adult form, they would form worms like this. That's one type of worm. Moss animals are looking like this. The hydroids, relative of the jellies, are looking like this. And then we have mussels. Everybody probably recognizes mussels. But of course, the big question is, did we find sponges? The answer is yes, but unfortunately, and I'm talking larval sponges, it was the bath sponge type of sponge that we found, not the glass sponge. We are still hoping to repeat this experiment with lessons learned from this one, but nonetheless, again, more information to provide to Department of Fisheries and Oceans here through this publication. So hopefully you have seen the idea on a larger, bigger picture framework of the incredible work and benefit that the sponge reefs are in terms of connecting the sponge reefs on the bottom with the plankton in the surface waters, and how important that is to basically our benefit. And the fact that having used those artificial trees demonstrated that larvae are in the water column, planktonic organisms, and they do and will settle out on the sponge reef. Those trees were installed, if not right in the reef, inside the reef, very close to it. Okay, however, now that you know that the skeleton is made of silicon dioxide, which is similar to glass, but not quite the same, they're sensitive. And they are sensitive to breakage by anchors, by downrigger lines, especially the lead weight at the bottom, or just smash through these. We have lots of evidence of that. And we've even seen sponges that have been sliced literally like a knife in two from the line. When I was going through the channels of the wall of glass sponges and the large, starting large and going smaller and smaller, and mentioning the whole concern about clogging, things like trawling, where a net is pulled across the bottom of the ocean, of the seafloor, sediments are resuspended. And when that happens, and it's near a sponge reef, and these sediments have been known to travel as far as 5 kilometers through the water, So it means that any sponge reef in the area could be affected. Mind you, this is not as big an issue in Howe Sound as they are elsewhere, like the Hecate Strait area. Traps, whether they're prawn traps, crab traps, they end up looking like this if they're dropped right into the middle of the reefs. This is an infraction. You can see how the sponge was smashed through and even hauled up in this trap. Another situation that has changed is that old traps never really they keep on fishing is what they do. You'll get, whether it's crabs or fish or whatever, going inside the crab traps, and then they pull in other organisms that want to feed on them, and unfortunately they generally can't get back out. So we call that they're just continuing to fish. Modern day traps tend to be made of the material that does eventually decompose. I don't know what the time frame of that is, but a little bit more environmentally friendly. Then we have the chemical contaminants that would have quite likely influenced the growth of the sponge reefs in Howe Sound. The wood fiber mill, 1964, and although it has been tremendously upgraded in terms of its virtually no emission of contaminants, prior to that, everything basically went into the water. And that would have, as with copper, would have affected the tissue, specifically of the sponges. And the tissue, primarily the tissue. Now the copper mine has closed down, and I'm sure many of you who live around the Sound have seen the increase in whales and dolphins, herring. That's a slightly different story, but nonetheless, there has been a general return of diversity of marine life into Howe Sound, and that is a really good thing. It's taken a long time, but we wonder whether or not the reefs, sponge reefs, were affected in those decades as well. Drilling for oil is another consideration. We don't have to worry about that here. Hecate Strait might have to, and I forgot to put in hydro lines. That we did actually communicate with BC Hydro to give them a better route to lay a cable from the mainland across to Bowen Island, actually, a number of years ago. So that's really good news that there's good collaboration that way. So hopefully through all of this, we've learned that they are rare. Less than 10% of all the sponges in the world, the glass sponges. They're fragile because of that silica skeleton. The spicule is made of that silica. They're vulnerable to sediment turbidity. So any of those types of fishing gear, even traps that are landing where they shouldn't, will resuspend sediments that could potentially clog these sponges. And if they can't pump, they're not getting oxygen, they're not getting food. So if that happens for a long period of time, they potentially would die. Now, the other thing that is an influence here to the survival and how sensitive these sponge reefs are, they're slow growers, maybe 1 to 9 centimeters per year. Usually it's faster the younger the sponge is, but it slows down the older they get. But we're looking at least 3,000 years for it to reach a 12-meter height. That may seem fast, but it's not. We don't really know how old these sponges get, the glass sponges. Estimates for bass sponge types is around 220 years. So you can imagine from this that if you are smashing and destroying the sponges within sponge reefs, if not the reefs entirely, they're not coming back tomorrow. They're not coming back anytime fast, and we really don't know how long that would be. So then you need to be thinking of that pumping capacity, the movement of water, refreshing and moving water 4 times a year in the Strait of Georgia and in Howe Sound, what that effect would have on all of the other marine life that we've just gone through. And those were only examples or a portion thereof. If they are damaged physically, biologically, or chemically, their recovery will be slow regardless. Okay, just there are many strategies that DFO has used, policies that encompass these sponges, or sensitive benthic organisms like sponges and coral. The federal government has bought into the law of the sea with the biodiversity. I'm going to not get this right, just give me a second here. Yeah, the United Nations Convention of Biological Diversity to protect and encourage diversity. That's become well known as being really important to keep these ecosystems and habitats viable. They have bought into the ACHI, Biodiversity Target 11, which is, again, trying to conduct responsible fishing practices and to encourage biodiversity and the growth of biodiversity. And in doing so, the federal government came up with a conservation targets. But I just wanted to go through this. The goal of this strategy was to conserve the health and integrity of Canada's Pacific oceans and ocean cold water coral and sponge species, communities, and habitats as integral components of a healthy and productive ecosystem, providing for economic and ecological value and sustainable use. So important, sustainable. And so what is incorporated in here is conservation, management, and research. The work that MLSS has done has helped DFO in terms of determining conservation value, let's say it that way, and how to best manage it then. So lo and behold, with all of that work and contributions MLSS made to DFO to help them in their efforts to determine conservation necessity, 9 marine refuges were established. Those are in red and in squares, you could say here. So you won't see the 12 that Glenn had initially identified because they had been combined into what we sometimes call complexes. Nonetheless, 8 marine refuges were established in 2019, and a huge thanks goes to Dr. Anya Dunham, who is at the Biological Research Station in Nanaimo, the chief research scientist behind all of this, and also Illyria Ladwick, who is, I think now, has always lived in Gibsons, for her support in ecosystems approach officer, as ecosystem approach officer with DFO. Very supportive, very cooperative with us in terms of what we were wanting to do or needing to do to help them. You'll notice gray areas here, Carmela Point, down here in Collywood Channel, Alberta Bay, September Morn here, and a couple of spots here. Now, this number of reefs here at Passage Islands had been discovered previously, but there were other areas that MLSS noted that had not been noted by Kim Conway. Oh, sorry, I forgot that one up there. Anyway, so they were not captured within this marine refuge in 2019. All of the ground proofing had been done on them, but it was kind of at a period where DFO had a very large turnover of staff, and they are all getting very much up to gear now. But let's deal with the marine refuges first. So discouraging fishing and anchoring in these marine refuges. They do have what we call a buffer. So there's the reef itself, and then a 150-foot, oh dear, is it feet or meters? See here, I get confused again. No, does it say on here 150? Oh dear, sorry, I'll have to find that. But there is a buffer zone around the actual sponge reefs as a protective area. So no bottom contact fishing. That includes traps, whether it's prawn, crab, no trawling, no groundfish hook and line, no use of those downrigger gear, and to please try and stop, avoid anchoring. So here's your healthy reef, and here is your damaged reef. Now, thankfully, just this year, DFO did give a status of fishery closure to those 5 purple areas that I had mentioned before that had not yet been protected. They are just fishery reefs at the, fishery closures at the moment, because they're still being worked on with the First Nations, coastal nations around the Sound, as to food, ceremonial food. Sorry, losing my brain. Food, ceremonial, and ceremonial and sustenance. Sorry, it's. I'll think of it. Anyway, on we go. So that's all good news. What's not necessarily good news is that in spite of the fact that these reefs and the marine refuges will go towards the federal government commitments, I and MLSS have some reservations. So let's just note these the targets for conservation strategy by the federal government was 5% by 2017, 13.81% by September of 2020. So in fact, the first marine refuges that were established fell into that category. New goals were set for 25% by 2025 and 30% by 2030. Okay, all sounds good, but is it working? Is it too late, too little, too late? First of all, marine refuges, although they are generally considered as a 25-year period definition as a marine refuge, fishery closures are only for 1 year and have to be renewed annually. So they're not permanent, okay? In the past couple of years in particular, we have had very noticeable violations directly within the reefs. So even if they're called marine refuges or fisheries closures or marine protected areas, something is not working. Our divers have been able to go down on a number of occasions, not as frequently as we had in the past, but certainly have noticed damage that is either caused by downriggers, by vessels anchoring to fish, dropping prawn trap lines, fishing lines, downriggers. Oh, sorry, I already said that. All of which we have evidence of. The damage that we're seeing in the past couple of years, we do not know if that is one or a combination of all three. nonetheless, these are all prohibited fishing apparatuses within the reefs, within these marine refuges and fishery closures. Okay, so we have residents who are and have reported vessels, in this case, very disappointing commercial vessel. I'm not, obviously it's with a telescopic camera set up, but we have license number, we have crab traps on the vessel, sorry, prawn traps on the vessel, and then the vessel without them, and then the vessel with them again. These are definite infractions of the marine refuge status. Deep down on Halkett, the map, one of the maps that I'd shown you earlier, you can see these glorious reefs of just those two different species of sponge, but look at how dense they are. That's live. We've got some dying ones here. That's okay, that's normal. That's what's helping to build the reefs. But each of these circles is identifying areas to look at. And unfortunately, because we had no idea that some of these infractions or violations were occurring, we thought there were problems because we were certainly seeing vessels out there, but we had no idea within the reefs that it was occurring. And this is what it's slightly different perspective, unfortunately, but like I say, we were caught off guard and didn't have a chance to record it properly, but you can see the damage that has been done, particular on the same Halkett Reef. So, and this is just in 2021. This is when the reefs were actually protected. So what is not working here? I want to say also before we go anywhere that with particular being out on the water, which Glenn is a lot in his vessel, he has come across recreational fishers right dead center with lines over the side right down into one of the sponge reefs. Glenn made an effort to communicate with these people, and apart from angry responses, et cetera, the final, I think, phrase that is important to know is they said, well, if the commercial fishers can fish here, why can't we? So that, I think, helps to accentuate how much of a problem this is. Maybe it's been accentuated by the pandemic and people wanting to get out on the water, out of their house, et cetera, but it's not new. It's not new. The reefs are now being considered and actually qualify under the requirements for something called the other effective area-based conservation measures. However, what's lacking at the moment is that there is no framework, no legal framework identified yet. So we're still in an awkward position. They're protected, but they're not really protected, and they don't have the legal backing to make these the enforcement really mean something. So we have talked to DFO, we've had meetings with them, we have written Honorable Minister Bernadette Jordan when she was in office. We need more officers. I feel very sorry for the fishery officers. They've got a large space to cover. And they're run off their feet, but what we need is more officers on the water. We heard just recently, this week as a matter of fact, there are going to be 4 positions hired for fishery officers. 2 will be in the northern region, 2 will be in the south, and we are very optimistic from this recent conversation that 2 will be in the Howe Sound Area 28. So Howe Sound, Vancouver Harbor, Indian Arm, and potentially down to Steveston as well. But that was the problem. The area that these officers are trying to cover is all the way from Squamish up to Seashelt, across to Galliano Island, and all the way down to Steveston. That's a huge area to cover. And there's everything from illegal crab traps being laid out. The sad part is that it's repeat offenders. It seems to be repeat offenders. One of the examples, I think, the fisher person dates back to something like 2009, and DFO has gotten a lot stronger in its fines and its lots of, whether taking their boat, they'll take their license. It depends on the circumstance. But when you get repeat offenders, actually one of them, who I think was off of Seashelt, is banned for life from ever owning a fishing vessel, not allowed to brokerage any fish, not allowed to be on a fishing boat, and the very next day was back out on a boat fishing again. So, it's like there's always a couple of bad apples in every barrel of apples, basket of apples. But it doesn't matter, because at the end of the day, it's the reefs that are suffering. It's all of the marine, sorry, I shouldn't use the word suffering. It's the reefs that are being damaged and broken. And all of the marine life that depended on it are now having to find other places, if there is, to go, or they will die as well. So is it too little, too late? I hope not, but you saw how long it takes for sponges to grow. It's not overnight. It's a long period of time. They've been around for millions of years. Think about it. It's not just a Canadian treasure. These glass sponges are a world treasure of an ecosystem. So hopefully you can gather, some of my passion, and certainly that of other MLSS board members and divers. These are worth protecting. And rather than seeing this, we would far sooner see this. So we are very hopeful that our new, or about to be new, fishery officers, who, by the way, probably won't be in place for another year, 2023, what's going to happen between now and then, but at least it's promising. And we are very, very thankful for DFO, federally and provincially, coming up with the funding and the positions, but enabling this extra enforcement, such that we hope that we will see more of this natural reef. Okay, that's it for me. I'm happy to take questions. I hope I haven't gone over too much. I will stop sharing. Or actually, I can take questions even now, Emily, if you want, in case I have to back up.
Yeah, if maybe if you do stop sharing your screen, I just have the screen to pop up again.
Right, there we go.
Don't mind, thank you. And then, yeah, if people have questions, we just encourage you to put them into the chat box, and then I'll just read them out to Sheila as they come in. Okay, so Sheila, from David Marmarick, he says, is it possible to have the protected areas marked on the GPS maps that sailors and fishers use?
Yes, it is. And in fact, oh gosh, there's a particular software that you can download and find that. You should also be able to get it through not the GPS. I was going to say you definitely need to go to the DFO website. Oh gosh, I'm sorry. I should have reminded myself of what that particular software was called, but most fisher people have it anyway, regardless of the sponges. So if I think of it, I'll send the information to Emily. But I do want to say that sometimes people who go out on the ocean don't realize that fishing is actually a responsibility. It's not up to DFO to, they're trying to regulate and maintain sustainable fisheries for everyone's benefit, but it is actually the responsibility of individuals who want to fish to learn about what they can and can't do. And that definitely has to be the DFO website. It's a bit convoluted and a little bit hard to follow at times, but it is your responsibility. So that's something to keep in mind, because sometimes we come across people that have no idea about that at all, and you need a license.
Great. Okay, so we have next question is, how was it figured out that the glass sponges filter that much water? All of the Salish Sea 4 times a year.
Yes. DFO did that, and in one of the areas that DFO actually did some experiments of their own using an ROV, because remember that's really deep there. They did dye studies. So they actually used the ROV and had select locations where they inserted dye. I believe it was around the exterior, on the outer walls of sponge, to see the time that it took for these little color cells to pull the dye in through the wall and out through the aperture. And they obtained that kind of information. I don't know what that was specifically, but they used that. And then using the diameter of the osculum, where the water is being pumped out of, and estimated from reefs, the size of the reefs, because that's part of ground truthing. They get an idea of how large the actual footprint of the reefs are. They calculated that number of 17 billion liters per day.
Great.
Yeah, hopefully that helps.
It's a great fact that I think really draws people attention to the importance.
Yes, and actually, sorry, I'll just throw in that 2 of the board members came up with the amount that those same sponges were pumping in a day is equivalent basically to all of the waste material that Metro Vancouver accumulates in one day from all of us living here. That amount of water is what these sponges are pumping through in one day. So if you think about that, that brings it a little bit closer to home.
Colin Loving, I'm having a little bit of a hard time deciphering your question. I think there's a few typos. Maybe you could type it out for me again. That would be appreciated. I'll just move on to the next one for now. So the next question, if sponges are filtering water, are they also threatened by poor water quality? I'm sorry if you said it and I missed it. I was just eating dinner.
That's understandable. So yes, poor quality. And that poor quality can include mining industries, as with the Britannia copper mine, copper coming into the water. Fish will even die from copper, so it no doubt would have damaged the tissue of these sponges. And again, the sediments, that's not necessarily poor quality water, but yes, any kind of contaminants in the water are quite likely going to affect the sponges. Sponges tend to be quite deep, so it does depend on whether those contaminants make it to those levels. But don't forget, these reefs are moving huge volumes of water. So chances are, if it's in the surface waters, it'll end up at the bottom where the sponges are as well.
Great. Okay. And then how will these new closure areas be communicated to the public, if you know? Are there going to be like increased notices in print and social media, or notices at docks? What's usually done?
Yeah, okay, so education is a huge aspect of this. It's an MLSS has been trying to do it with what we call beach interpretation programs. We've had those in Porto Cove, on Bowen Island. Would like to have done them at a few other places, like the camp, Elphinstone Camp, is it, near Gibson's? Yeah,
Elphinstone.
Yeah, we were unable to do it then, and we haven't been able to do them for the past couple of years, but that's one way of educating. With this recent meeting with DFO, they are definitely coming up with some very large signages that are going to be placed at strategic locations within Howe Sound that boaters, I don't think, will be able to avoid. We have noticed that fishers will vein that, you know, they didn't know that they were refuges, but like I say, or even reefs before the refuges were formed, they will sometimes just plead ignorance, but that doesn't count, because it is, as I said, fishers' responsibility to know what they are fishing, to get a license and know what's entailed with having a fishing license. So it is up to individuals to find that out. And again, I'm sorry, I can't remember the name of that software. That is what fishers can download, but most fishing boats have GPS, and they should be able to access the areas of these refuges through that software. Okay.
Okay. So I'm just going to read Colin's question out. He says, "Thank you, Sheila. I noticed few, I believe, inside the sill of the sound. Is this perhaps because more searching is needed, or perhaps sediment from the Squamish River, or the periodic low oxygen here? Any comments appreciated?"
Yes. Okay. So the initial, great question, actually. The initial reef that was found near Defense Islands by Kim Conway and his crew, so up near the sill into the Squamish River, and a number of other reefs were found around there. One of them was essentially dead. So we think, and this is not just MLSS, but I'm trying to remember if it's Sally Lee's or DFO researchers that are wondering if that dead reef near Defense Island, which is closer to the sill, why it's dead. It is slightly, it's probably one of the shallower reefs, but it is also very near to all of the outflow of the Squamish River, where it could well have been influenced by wood fiber or Britannia mine. So there is some, you know, questioning, is that a possible reason why that particular reef is dead, and will it ever recover? We don't know, because surface waters, again, are also warming more with climate change, and there's concern about that for various reasons as to the impact that it will have on the reefs, even at depth, let alone at that shallower reef close to the sill. So that's yet to be determined. I'm not sure, you know, exactly when we can get things together to actually focus on that so much as other things that we want to do at the reef that may be of more value. Anyway, hopefully that gives you some response.
Well, I encourage Sheila, I think that's the last question we have. I really encourage you to read through the chat because lots of people left thank you messages for you in there. And I myself, and on behalf of the Marine Education Center here in Gibsons, would also love to thank you for your time and your passion and commitment to the Howe Sound at Gibson and to the glass sponge reefs. We've learned a lot this evening.
Thank you, marvelous.
I also just once again want to thank our presenting sponsor, the Sunshine Coast Foundation, for supporting this talk and the 3 Ocean Optimism Speaker Series events that came before it. And as it is our last time connecting with our audience, if you want to continue to support us, we encourage you to continue to participate in the programs that we offer here at the center. If you have kids, grandchildren, we do camps through spring break and summer. We do school programs. We have after school science club for high school kids. We do lots of different family activities throughout the year. So keep in touch with us. We also take on volunteers from 12 years old into senior lifestyle. There's lots of different ways you can get involved with us. And again, as we are a charitable organization, if you are able to support us financially, we encourage you to donate. If you go to canadahelps.org and you search our name, we come up as a charitable organization there that you can donate to. And that link is also connected through our website, which is www.gibsonsmarine-ed.org. And a great way to stay in touch is to follow us on social media, so Facebook or Instagram, and to sign up for our newsletter, which we send out once a month to kind of let everyone know what we have going on. And this QR code on the screen, if you hold your phone up to it, it will take you to the link that you can sign up for our newsletter if you're not signed up already. That's kind of it.